Hello, and welcome back to the Mechanochemistry discussions. This seminar series is hosted by the NSF-funded Center for Mechanical Control of Chemistry, the CMCC. I'm Ashley Martini. As always, the goal of this seminar series is to bring the community together. Our seminars stream live on the third Thursday of every month, in today's case, the fourth Thursday of every month, and are also available to watch anytime on the YouTube channel. We've had a great group of speakers so far as part of the seminar series, and again, these are all available on YouTube. We are also looking forward to some seminars in the months ahead. We hope that you will join us for all of them. Before we get started, a great big thanks to a few folks. First, Dr. James Batiste, the Center Director, Jennifer Belsick, the Center's Administrative Coordinator, and two CMCC students, Katie Floyd and Quintarius Moore, who are invaluable in the production of the seminar series. Thank you again for joining us. Please do follow us on the YouTube channel as well as on Twitter. And lastly, a few quick guidelines. Reminder that the seminar is being recorded. If you have any questions for the speaker, you may either email them to us at cmccdiscussions@gmail.com, or you can post them in the YouTube channel. Either way, they will be propagated to the speaker at the end of the presentation. Finally, last but not least, it's my pleasure to welcome today's speaker, Professor Max Robb. Professor Robb was born and raised in Colorado and obtained his bachelor's in chemistry from the Colorado School of Mines, where he began research in synthetic polymer chemistry under the guidance of Professor Daniel M. Naus. After graduating in 2009, Professor Robb carried out his PhD studies in the laboratories of Professor Craig Hawker at the University of California, Santa Barbara. His doctoral research focused broadly on the synthesis of functional organic materials and was recognized by the American Chemical Society with the 2016 Henkel Award for Outstanding Graduate Student in Polymer Chemistry. Professor Robb conducted postdoc work with Professor Jeff Moore at the University of Illinois Urbana-Champaign as a Beckman Institute postdoctoral fellow prior to joining the Division of Chemistry and Chemical Engineering at Caltech as an assistant professor of chemistry in September 2017. Research in the Robb group seeks to advance the fundamental understanding of mechanical force transduction at the molecular level and develop strategies to create force-responsive molecules and functional materials. The group's research has been recognized by a number of awards, including the Beckman Young Investigator Award, the Sloan Research Fellowship, the NSF Career Award, Camille Dreyfus Teacher Scholar Award, and the PMSE Young Investigator Award. It is my pleasure to welcome Professor Max Robb.
All right. Well, it's a pleasure to be here today and contribute to this mechanochemistry discussion through the CMCC. So thank you to the organizers for the invitation. I know that mechanochemistry is a pretty broad field. And so what I'm going to tell you about today is the work that goes on in my lab at the California Institute of Technology, where we're really focused on this area of polymer mechanochemistry. And I'll share with you some of our work over the last several years. And so I would like to start out with just a sort of brief historical perspective on the field. In order to do so, we have to really go back to sort of the early days of polymer science, really back to about 1930s or so, where it was understood that if you take a polymer chain and you subject it to stress, that polymer chain will undergo a bond scission reaction, typically thought of as just a bond homolysis. And so, this process was almost always considered a degradative process. So you cleave those bonds in a polymer chain and you essentially degrade the mechanical properties of your material. In the 1980s, this work from Encina showed that you could actually program into a polymer some mechanical responsive behavior. And so what he did was very clever and incorporated these peroxide linkages, these mechanically weak peroxide linkages into a polymer chain, and showed that by doing that, you could actually modulate and increase the rate of polymer scission through rupture of those weak peroxide linkages. Going forward to 2005, there was this seminal paper that came out of Jeff Moore's lab at University of Illinois that took this one step further. And what he showed was that if you incorporate a weak azo linkage in the center of a polymer chain, subject that polymer to mechanical stress, you can site-specifically cleave that polymer at the location of that weak azo linkage. And so this really sort of built this idea of a mechanophore, which is a term that I'll use throughout my talk today. And what I mean as with respect to a mechanophore is just a stress-sensitive molecule that exhibits selective reactivity under force. And so this was a huge step forward. But I would say that it wasn't until a couple years later where the field really sort of emerged. And we're able to now harness these mechanical forces in polymers to not only do polymer degradation, right, but to actually do really interesting and productive chemistry. And the paper that I want to highlight here, that again came out of the Moore lab in 2007, was this fascinating approach where they showed that using mechanical force, you could promote a very unusual ring opening reaction of benzocyclobutene. And more specifically, what they showed was that if you take the trans disubstituted isomer of benzocyclobutene and the cis disubstituted isomer of benzocyclobutene, and you apply mechanical stress across that molecule, the cis isomer undergoes a formal disrotatory electrocyclic ring opening reaction to form this EE isomer, which is the same product that you get from the from the trans isomer. And so what this showed is that using mechanical force, you could actually bias reaction pathways and access reactions that are otherwise inaccessible. And so this was a very interesting transformation and discovery. A couple years later, through computational approaches, the Martinez lab showed how this reaction is actually occurring. What they showed is looking at the cis isomer of benzocyclobutene. As you increase the amount of force applied to this molecule, you systematically change the shape of the potential energy surface such that at a sufficient amount of mechanical force applied to that molecule, this reaction actually becomes barrierless. Right? And so through this process, which we now refer to as a force-modified potential energy surface, we can understand how mechanical forces are able to sort of perturb molecular systems and in many ways access these very unusual chemical transformations. Over the years, a number of very interesting chemical reactions have been discovered in this field. One that I like to point out is this work from the Stanford group. This was work led by Jan Shaw, Noah Burns, and Todd Martinez, where they showed that you could take these very unusual and interesting polylaterine materials and essentially unzip them using mechanical force to generate these conjugated polyacetylene materials. So essentially converting an insulating material into a semiconducting material. And finally, I'll point out an area within polymer mechanochemistry that I think is particularly important, which is the development of so-called molecular force sensors, or mechanophores that undergo a visible change upon mechanical activation. So shown here is spiropyran. It's a well-studied mechanophore that undergoes a 6-pi electrocyclic ring opening reaction under force to generate these colored and highly fluorescent merocyanine dyes. Another example are these dioxetane mechanophores developed by Rintse Besma's lab in the Netherlands that actually undergo a chemiluminescent reaction upon mechanical activation. And so, again, these are useful for developing materials in which we can actually visibly observe and visualize mechanical stresses at the molecular level in those materials. And so what my lab has focused on is really the molecular design and the development of new mechanophores and sort of molecular design strategies for coupling mechanical force to these covalent mechanochemical transformations. And so I'll tell you a few stories today. The first will focus on a strategy that we've developed over the last several years, which relies on mechanical gating to access a variety of different sort of functional responses in different mechanophore systems. The first little vignette that I'll tell you about, or a couple of vignettes rather, is on this idea of mechanically gated photo switching, which presents an alternative route to designing materials that change color under stress. And then I'll show you how we've used the same general strategy to design molecules and materials that, upon mechanical activation, release a covalently bound small molecule payload. In the second part of my talk, I'll tell you a little bit about our work on the development of Naphthopyran mechanophores that are mechanochromic and undergo this color-changing reaction under stress, and some really unusual chemistry that we've discovered within that particular area. And I won't highlight this too much, but something that's pervasive in all of the research that we do in my lab is really the development of fundamental insights into force-coupled reactivity, how we understand molecular design principles, structure-activity relationships with respect to mechanochemical reactions, and also developing better analytical tools and perspectives that enable us to provide these deeper fundamental insights into reactivity. So I want to start with, the question, how do you actually design a mechanophore? And the molecule that I'm going to use as an example is this particular cyclopentadiene maleimide Diels-Alder adduct shown here. I'll come back to this and tell you why we were interested in this molecule. But our hypothesis was that with this particular motif, upon mechanical activation, this would undergo a formal retro 4+2 cycloaddition reaction or retro Diels-Alder reaction to unmask this latent diarylethene photoswitch, which we could then use UV light to induce a photoisomerization reaction leading to a color change. And so that was the hypothesis in our original molecular design. The next step in our process is typically simulation. And something that has emerged as a very powerful computational tool for us is what we refer to as COGEF, which stands for Constrained Geometries Simulate External Force. So essentially, this is a very simple computational method that allows us to predict mechanochemical reactions in silico. I'll point out that it was originally developed by Martin Bayer back in 2000 and has since become a very powerful tool for the field of polymer mechanochemistry. In this particular method, what we do using this example is we constrain the distance between those two methyl groups on our truncated model. And we simply increase the displacement between those methyl groups in discrete steps. And then at each one of those steps, we do a DFT optimization of the geometry. And by doing this process, we can predict what happens as you mechanically elongate this molecule in terms of its chemical reactivity. And so in this particular case, what we might expect out of the simulation is to see a retro Diels-Alder reaction, right, producing our expected diene and dienophile. Or perhaps what we might expect is some maybe carbon-carbon bond scission that is not desired, in which case we would go back to the drawing board and sort of reevaluate and redesign our molecule and subject it to this iterative process again. But once we have a computation that supports our original hypothesis, at that point, we would then go into the lab, we would synthesize that molecule, and then we incorporate it covalently into a polymer. And the polymer here is really important because it allows us, it's essentially our handle that allows us to transduce force to that particular molecule. Once we have our polymer, we would then do a series of activation experiments, either in solution phase or solid state. I'll talk about both today, and in order to characterize the reactivity that's happening upon mechanical activation. And so just to give you a sense of what these COGEF calculations look like, this is for that same molecule I just showed you on the previous slide. This is the COGEF plot. We're plotting relative energy as a function of displacement between those terminal methyl groups. And so as we go from left to right in this plot, we're basically just stretching that molecule out in discrete steps. And each of these data points represents one of those steps. And so you can see that we start from equilibrium geometry. And as we stretch that molecule out, we see this increase in energy of the system. Until we get to a maximum. If we look at the structure that's predicted at that point, perhaps you can see it, maybe you can't, but this molecule is very, very distorted, as you might imagine, right? If we just increase that displacement a little bit further, we see this huge drop in energy, right? And just on the other side of this discontinuity, if we look at the molecules that are predicted after this chemical reaction, what we see is actually that COGEF predicts the formation of this cyclopentadiene, this diarylethane motif, and a maleimide through what would be a formal retro Diels-Alder reaction. So one other thing that I would like to point out about COGEF is it's really good for qualitative aspects, right? We can predict what chemical reaction is going to occur as we mechanically activate a particular molecule or model. From a more quantitative standpoint, the other thing we can get out of this is actually a force or a maximum force associated with a transformation. And that's really just related to the slope of this curve immediately prior to this discontinuity. And what we've shown is that the magnitude of this force value actually gives a relatively good prediction of its mechanochemical reactivity. And so this is one of the features that we sort of take out of these COGEF calculations as well. I don't want to spend too much time on this, but I will say that COGEF has been used over the last 15 years or so, but in sort of niche areas and sort of odd applications. And so one of the things that we tried to do several years ago was really validate this COGEF method as a tool for predicting mechanochemical reactivity. And the way that we did that was essentially surveyed the entire literature for every mechanophore that had been reported up until that point. And we applied this consistent COGEF method to all of those molecules. So we ended up studying almost 130 molecules for which there was experimental reactivity data in the literature. And what we find is that using a relatively, say, unsophisticated level of DFT, which means that we can do these calculations relatively high throughput, we find that nearly 90% of our calculations predict the reactivity that is validated through experiments. And so this really showed to us that CoGEF is a good predictive tool, and it has been extremely valuable in our lab for being able to assess and predict mechanochemical reactivity and sort of influence our molecular design of different mechanophores. Okay, so the last step here that I want to talk about is how we actually apply mechanical forces to these mechanophores or to polymers. And I'm going to focus here on the solution phase experiments. So in solution, we use ultrasonication to generate mechanical forces on polymers. And the way that this works is that when we apply high intensity ultrasound to a dilute solution of polymer, that sound wave propagates through that medium and it generates a pressure wave. And this leads to something called cavitation. Cavitation is essentially the nucleation and the growth and then the collapse of these microbubbles in solution. And what that does is it creates these local shear environments, these shear fields. If you have a high molecular weight or a long polymer chain in these shear fields, it leads to this rapid elongation of that polymer. And if we have a mechanophore covalently embedded in that polymer, then we can essentially focus mechanical force to that particular molecule or moiety of interest. And so ultrasonication, we can apply very high strain rates, which is great. I would say, importantly, it's experimentally simple and very reproducible. But perhaps most importantly, it enables, because it's in solution, it enables us to use conventional analytical methods, things like UV-vis absorption, fluorescence spectroscopy, NMR spectroscopy, to really characterize the types of chemical reactions that are occurring under these mechanical forces. A couple of things to note is that under these conditions, force is maximized at the chain midpoint. And so if we can locate our mechanophore, potential mechanophore, at this position in the polymer chain, then we can essentially focus mechanical force to that particular molecule. Longer chains experience greater force. So this is basically our handle for being able to modulate how much force a molecule or a mechanophore experiences. And there's also a threshold chain length for activation. So what we see if we monitor reaction rate as a function of chain length, right, we basically see no reaction until we get to this threshold chain length or a threshold force, at which point we start to see an increase in activation rate with the length of that polymer chain. Okay, so now to talk about some chemistry. I'm going to start with this idea of mechanical gating and how we have used that as a strategy to design a wide variety, actually, of different mechanophores with very different function. And the first thing that I'll tell you about is how we sort of entered into this research, which is the development of this idea of mechanically gated photo switching. As I sort of mentioned earlier, we were interested in developing a different strategy that would allow us to couple mechanical force to a color-changing transformation. With the idea here being that if we actually decouple that functional response, this color change, from the mechanical activation event, it would allow us a very general and potentially modular platform for designing molecules and materials. The other advantage here is that we thought that if by designing this cyclopentadiene maleimide Diels-Alder adduct, which undergoes this retro-Diels-Alder reaction under force to unmask this latent diarylethane photo switch, right, this could really serve as a way to record the mechanical history of a material, right, in this particular process. And then after that mechanical event, we could read out that mechanical history through this photoisomerization reaction using light, right, which changes the color of this unmasked diarylethane photo switch. And so that's what I'll tell you about briefly. I'm going to sort of skip over our first generation design, but essentially what we showed was that if we have this cyclopentadiene maleimide Diels-Alder adduct shown here, upon mechanical activation, it does indeed undergo a formal retro-Diels-Alder reaction to generate this diarylethane photo switch. And then upon UV light irradiation, we can do this ring closing reaction to generate the colored isomer. Now, the reason I'm skipping over this is because while it worked, there were a number of challenges associated with our initial molecular design. And so what I'll focus on here is sort of a second generation of this system that accomplishes the same goal, but the chemistry is significantly improved. And so we've designed now this particular cyclopentadiene maleimide Diels-Alder adduct mechanophore, where we have polymer attachment at this nitrogen position, and then also now on this latent cyclopentadiene. Upon mechanical activation, it undergoes a formal retro-Diels-Alder reaction to generate this diarylethane photo switch. And importantly, now what this means by moving that polymer attachment handle up onto this cyclopentadiene motif, it allows us to now install a variety of different aryl groups on these thiophenes, which allows us to essentially modulate the photophysical properties of that masked photoswitch. And I'll tell you a little bit about that chemistry. And so skipping some of the development here, a student in my lab, Ross Barber, developed a really elegant and robust synthesis of this particular diol. And so starting from here, what we can do is we can take advantage of these thioneal chloride functional groups, and we can do a palladium catalyzed cross-coupling reaction at this stage in the synthesis to install, again, a variety of different arenes at this position to modulate the photophysical properties of our masked photoswitch. We can then, following a sort of conventional protocol that we use in our lab all the time, we can take this diol and we can functionalize these alcohols by installing alpha bromoester functional groups through the reaction of this particular molecule. And then we can use that as a bis initiator to do a living radical polymerization, in this case of methyl acrylate. And so what we're left with at the end is a linear polymethyl acrylate polymer that contains near the center of that polymer chain our cyclopentadiene maleimide Diels-Alder adduct or potential mechanophore. And so here I'll just show you sort of the functional response, functional reactivity of this particular polymer and this mechanophore. And so again, what we expect is if we take our polymer and we dissolve it in solution, we can subject it to ultrasound. And that mechanical force should induce a retro-Diels-Alder reaction to reveal this unmasked diarylethene photoswitch. It should remain colorless. And then if we shine UV light on that system, it should undergo photoisomerization reaction to generate this colored form of that diarylethene photoswitch. And the molecule that I'll show you the data for here is the phenyl substituted derivative. And so here we're going to look at the changes in UV-vis absorption as a function of these different steps in this process. So here is the UV-vis absorption spectrum of just our starting polymer before we do anything to it. And what I'll show you is that if we take that polymer and we just irradiate it with UV light, we see absolutely no changes to the absorption spectrum and no changes to the color of that material. However, if we activate it mechanochemically, we see some small changes in these short wavelength region, right? But if you look at that solution, which is actually here, and then actually any of the solutions of any of our derivatives, they remain completely colorless after that mechanochemical activation event. But now if we take those solutions and we irradiate it with UV light, we see this color changing response, right? And specifically for this phenyl substituted derivative, we see the appearance of this visible absorption band at about 550 nanometers, right, corresponding to this pink colored solution. So again, this is a mechanically gated photo switching reaction where mechanical force is required for this color changing reaction to occur under light. I won't show the data here, but one of the important things that we do a lot of control experiments in my lab, and one of the important controls that we always do is to confirm that the reactivity that we're seeing here is actually due to mechanical activation. And the way that we do that is we design a polymer that instead of incorporating our Diels-Alder adduct, in this case, near the middle of the chain where force is maximized, we locate it at the very end of the polymer chain. And the end of the polymer chain under these conditions experiences no force. And so by subjecting this polymer to the same sequence of experimental conditions, what we see is no changes in photochromism for this particular molecule, confirming again that this response that we see here really is due to mechanical activation of our mechanophore. The other thing that I'll just point out briefly is that we were able with this system to extend the properties and the reactivity that we observe in solution to actual solid state materials. And so we showed this through cross-linked polymethyl acrylate networks, that upon compression of those networks that were cross-linked through a mechanophore, we also observed this mechanically gated photo switching reaction leading to a color change in our sort of active material and no color change in a control material in which mechanical force cannot be transmitted across that molecule, that mechanophore. So this was a really nice system, and arguably the chemistry was really nice and worked really well. But I want to point out one thing, which is that despite the increased modularity that we designed into this particular system for mechanically gated photo switching, it still requires 3 steps for each polymer that's synthesized from our sort of parent mechanophore, right? And so if we wanted to make 4 different polymers that all changed 4 different colors upon mechanical activation, that means that 12 reaction steps essentially would be required to generate that library of materials, which is still not ideal. And so one of the things that we've been thinking about for a long time in my lab is there a way to design a single parent polymer that we can mechanically activate to generate a wide variety of different dyes or different photo switches, again, from that single parent material. This sort of got us thinking about this other class of photo switches, which are referred to as donor acceptor Stenhouse adducts. I'll tell you a little bit about those. So this is a DASA molecule, and it undergoes what's referred to as a negative photochrome, and so it undergoes this ring closing reaction under visible light to generate this colorless state, which is reversible thermally. What we're really interested in about this class of molecules, though, is one, how easy they are to access synthetically. And so we can just simply take furfural and a variety of these different so-called carbon acid derivatives, and that generates this activated furan precursor. You can then just treat this activated furan with a secondary amine, and that generates this DASA dye or this DASA photo switch. And so what caught our attention with this chemistry was that just by changing the identity of this amine used in this step, we can completely change the photophysical and photochemical properties of that DASA dye or that DASA photo switch. And we thought actually this was a really nice opportunity to use our mechanically gated photo switching strategy, where we could essentially intercept this activated furan intermediate and mask it in the form of a mechanically active molecule or a mechanophore. And so the system that I'll tell you about is this particular heterodiels-Alder adduct. Upon mechanical activation, it undergoes this retrodiels-Alder reaction to generate that activated furan intermediate in a material. And then what we can do is just treat it with a variety of different secondary amines, and each of those will lead to a unique generation of a unique DASA photo switch. And so this is some of the chemistry. The chemistry actually turns out to be very straightforward. We can take this particular activated furan species that has this terminal alcohol. You can access this in just 2 steps from commercial materials. And we can do this heterodiels-Alder adduct with this particular vinyl ether to generate our heterodiels-Alder reaction to generate our heterodiels-Alder adduct here that has, again, those terminal alcohol groups. And so following the same sort of principles that I showed you previously, we can then functionalize those alcohols, convert them into alpha bromoesters. And now we have our bis initiator for radical polymerization, where we can grow, in this case, polymethyl acrylate chains off of each of those sites to generate a polymethyl acrylate polymer, where near the middle of that polymer, we have our heterodiels-Alder adduct. So the first set of experiments that I'll show you about is using ultrasound to examine the reactivity of this particular heterodiels-Alder adduct. And so if we take our polymer, we subject it to ultrasound, again, it should undergo this retrodiels-Alder reaction to reveal this activated furan intermediate. And then if we just treat that with a secondary amine, we should be able to form a DASA dye or a DASA photo switch. And so here in this plot, what I'm showing in the yellow trace is this polymer after mechanical activation with ultrasound. And the dashed trace is the absorption spectrum of a small molecule reference compound. And so I'll show you a variety of plots. The dashed trace is always a small molecule model, either this activated furan reference or the actual DASA small molecule as a comparison. And then in the picture on the right-hand side is the solution of our mechanically activated chain-centric polymer, and on the left is our chain and functional control. And so here, after activation, we see the generation of this nice yellow color, and it matches the absorption spectrum that we would expect based on the generation of this activated furan. So now we can take that mechanically activated ultrasonicated solution, and we can just throw in some amine. In this case, we add in some diethylamine, and we see the conversion of that nice yellow color into this nice pink color, and the generation of a DASA photo switch. Now, using this same parent solution, we can just simply change the amine that we add. Now, in this case, we can add this aniline derivative, and we can now generate this nice, beautiful purple colored DASA photo switch. And we can do it again. We can take that parent solution. In this case, we can add in some indoline, and we can generate this nice blue colored DASA photo switch, again, from that same parent material that was mechanically activated. And so we were excited about this. And what we wanted to do next was translate this into solid materials. And to do that, it's actually relatively straightforward. we can take our same diol functional mechanophore, and instead of installing those alpha bromoesters, we can install these esters that have these terminal olefins. And then we can just use hydrosilation chemistry to incorporate these covalently into silicone elastomers. So very, very simple chemistry, and we can access really nice materials, again, that covalently incorporate our mechanophore, or a control molecule, where in this case, we only have one site of attachment. And so in this case, our control, again, mechanical force cannot be transmitted across that Diels-Alder adduct, and we would expect this to not exhibit any mechanochemical reactivity. So if we take that active PDMS network that contains our cross-link mechanophore, we can stretch that, and then we can release it. And in this experiment, essentially, we didn't see anything happen. But if we take that material that had been mechanically activated in tension, and we just dip it in a solution of this aniline, we see this relatively faint, but nevertheless, I think clear coloration, this blue coloration that appears in the gauge region of this material where mechanical force is maximized, right? Indicating that we have indeed formed, through this mechanically gated DASA formation, a DASA photoswitch through this chemistry. And then upon visible light irradiation, these behave just like we would expect any DASA photoswitch, where we basically see the disappearance of that color under visible light. And I'm not showing that here, but this is thermally reversible in the dark. And so we can form DASA photoswitches now through this chemistry in solid state materials. And that sort of led us to this final demonstration that we're really excited about, which is basically the ability to use this chemistry to do multicolor lithography using mechanical force. And so I'll tell you sort of schematically, our idea for this. If we take now a piece of a film of this active silicone, right, this elastomer, now we can apply localized force through compression using a stamp, so an embossed stamp that has this sort of flower petal pattern that will become apparent. And when we do this, we should only see that formation of that activated furan intermediate in the region of local compression in that material where mechanical force was applied. Then we can globally treat that material with an amine and form that DASA dye or that DASA photoswitch, again, only in the location of the material that was mechanically activated. We can then just simply iterate this process. We can apply a different stamp, right, and a different amine, and then a different stamp and a different amine, ultimately to sort of iteratively grow this composite multicolor image, in this case, producing the image of a flower, right, using mechanical force. And so I'll show you how this actually works in the lab. This is a 2 centimeter by 2 centimeter, this is a 2 centimeter squared piece of PDMS that contains our mechanophore. We can then apply compressive force using this embossed stamp. And you can hopefully see the appearance of this sort of yellow color, again, only in the region of compression that matches our stamp. We then take this film and we just dip it in a solution for 90 seconds of diethylamine, and we form this nice purple colored DASA in the region of local compression. We can then iterate this using this sort of stem stamp to form the stem of our flower, and then treat this with indoline in the same fashion. And this produces this sort of blue-green colored stem of our flower. And then the center portion of our flower using the circular stamp, treating it subsequently with this aniline derivative to form our green center. And so ultimately sort of building up this multicolor image of a flower in this case. These are also photo switches. And so one of the interesting things about this is that now they exhibit this sort of 4D behavior where we can now print in 3 dimensions these dyes, and then also control their behavior in time, temporally, using light. And so we can sort of have portions of this material partially disappear and then reappear after their formation. Applying the same process, the same sequence of processes to our control film, of course, produces no colored image, as you would expect, confirming that this is a mechanochemical process that's responsible for the generation of these images. Okay, so I now want to sort of change gears, but show you how we can use this mechanically gated reaction strategy in a totally different direction, which is for the development of molecules and materials that lead to the triggered release of small molecules upon mechanical activation. And so in this particular case, what we've done is design a furan-maleimide Diels-Alder adduct, now incorporated into polymers, where upon mechanical force, it also will undergo a formal retro-Diels-Alder reaction. But now, instead of revealing a latent photoswitch, we reveal this highly unstable 2-furyl carbonyl derivative, or a furan derivative. And we've designed this in such a way that once it's revealed mechanochemically, it spontaneously will decompose through a very well-defined process, in this case through a decarboxylation cascade, ultimately releasing a small molecule alcohol, which is bound through this carbonate functional group. And so we were originally inspired for this chemistry by some work out of Elizabeth Gillies' lab in 2017, where she showed that if you had these simple furan-maleimide Diels-Alder adducts connected through this carbonate motif on these so-called self-immolative polymers, you could use heat to thermally activate that retro-Diels-Alder reaction, revealing this furfuryl carbonate motif, which upon subsequent heating will undergo that decomposition reaction, and in this case, ultimately completely depolymerizing the polymer because it's one of these so-called self-immolative polymers. I've sort of highlighted the fact that both of these steps, especially the second step here, required significant heating. And that was something that we wanted to get away from, if we were going to use this chemistry in a mechanically gated, sort of mechanically triggered small molecule release fashion. And so to do that, we turned to computation. And what we found was that we could modulate the activity of these furfuryl carbonates with respect to essentially the rate of that carbonate fragmentation reaction by changing the substitution on our furan. And so what I'm showing here is a series of computations where, again, we're looking at the activation energy associated with carbonate fragmentation here for just our sort of naked furfuryl carbonate, and then upon addition of a methyl group at the 5 position of that furan, and then finally a methyl group at the 5 position, but also this alpha methyl group, which actually turned out to be a key structural feature. As we increase the alkyl substitution on these motifs, we see that the activation energy decreases from about 29 kcals per mole down to about 22 kcals per mole, again, for that carbonate fragmentation reaction for this particular motif that has 2 alkyl groups. One, again, importantly, being this alpha methyl group that I'll tell you a little bit about. So we identified this particular furfuryl carbonate as a nice starting point to investigate this sort of mechanically triggered release chemistry. What I'll tell you about in terms of that furfuryl carbonate is we can actually isolate small molecules of these furfuryl carbonates. This is, again, analogous to that dialkyl substituted compound on the previous slide. And here we have attached to our carbonate, we have this coumarin motif. We specifically designed this as a model because this exhibits a fluorogenic response. So in the carbonate, this is essentially non-fluorescent or weakly fluorescent. But once it's released as the phenol, it becomes highly fluorescent. And so we can use fluorescence spectroscopy to track the release of this hydroxycoumarin molecule. But if we take this small molecule furfuryl carbonate, it's relatively stable in aprotic solvents like acetonitrile. But if we add in a small amount of a protic solvent like methanol, this will undergo this spontaneous decomposition reaction, undergoing this decarboxylation, ultimately producing hydroxycoumarin, and then going through this furfuryl cation intermediate, which is intercepted by methanol to generate as a byproduct this methyl furfuryl ether compound. And so if we monitor the kinetics of this transformation, we can see that with this particular substitution, we have a half-life of this decomposition reaction at room temperature of about an hour. And we have the reaction is very clean. So we see basically 100% release of our hydroxycoumarin compound upon complete decomposition. Just as a comparison, we've also done this experiment on those other 2 sort of less substituted furfuryl carbonates, right? This is the one most analogous to the one that Gillies had looked at previously. And then this furfuryl carbonate has that single alkyl substitution at the 5 position. Under these same conditions, both of these molecules decompose with a half-life on the order of about 3 weeks, right? So that alpha methyl substituent turned out to be a really key structural motif to influence the rapid decomposition of this molecule, meaning that once we activate our mechanophore and produce this latent furfuryl carbonate, it would decompose readily to release that small molecule. And so we can access a similar sort of bis initiator motif of our furan-maleimide Diels-Alder adduct that now has this secondary alcohol shown here. This is actually a pretty modular synthesis. We can access this on gram scale in just 4 steps. And then from this, we can install our cargo through the reaction of a chloroformate, right, to install this coumarin carbonate motif. And then we can, again, just use this as a bis initiator in a living radical polymerization to grow our polymer chains. And now we're left with, again, a linear polymethyl acrylate chain where near the middle we have our furan-malamate Diels-Alder adduct that has this cargo-loaded coumarin through that carbonate motif. And so we can apply the same sort of experimental conditions that I've showed you previously. We can take this polymer dissolved in solution. We can apply mechanical force using ultrasound. Again, that should undergo this retro-Diels-Alder reaction to reveal this latent furfuryl carbonate. And we've designed this in such a way, again, where we expect now the relatively rapid decomposition of this material to ultimately release hydroxycoumarin, our small molecule. And indeed, if we take this polymer and we subject it to 150 minutes of ultrasonication and we monitor the fluorescence response, we see this really nice fluorogenic response from our chain-centered polymer. And you can see that again in a photograph of that solution under UV light. Compared to a control material, this is a chain-end control in which we see essentially minimal or negligible fluorogenic behavior, confirming that mechanical force in this case can be used to effectively release hydroxycoumarin. So one of the challenges that we ran into with this particular chemistry initially was the fact that if we just replace our hydroxycoumarin with an aminocoumarin, so now we have this carbamate instead of a carbonate, about 15 days after activation, we see essentially minimal fluorescence response, indicating that we really have very little of this aminocoumarin being produced. And really that comes down to the stability of this intermediate, right? So we're producing this furfuryl carbamate under mechanical force, but it's just kind of sitting there. And so again, we turn to computation to examine a variety of different substituent effects on that intermediate furfuryl carbonate or furfuryl carbamate. And I'll show you just one. We've identified this particular one where, again, we have a alpha methyl group, so alpha to that carbonate or carbamate, but now we've installed an electron donating phenoxy substituent on at the 3 position of this furan, which we predicted and indeed shows that we can accelerate the release of that small molecule with this electron-rich furan now. And so going from, this is looking at the furfuryl carbamates, or the release of amine, going from sort of the parent structure, we have an activation barrier about 33 kcals per mole. With the installation of both of these substituents, now we've lowered that barrier to about 23.8 kcals per mole, suggesting that we should now be able to release things like amines on relatively reasonable timescales. And so if we look at that experiment, again, this is the mechanophore in our linear polymer with the phenoxy substituent and an alpha methyl substituent, and we subject it to the same conditions. We now see, this is the plot that I showed you previously for comparison. We now see immediately after sonication this nice fluorogenic response, and then within about 3 hours after sonication, we see essentially quantitative conversion of our unmasked furfuryl carbamate and the release of our aminocoumarin cargo. And so we showed that this particular mechanophore works very well for the release of not only phenols and arylamines, as I showed, but also alcohols, alkylamines, also things like sulfonic acids and carboxylic acids. And so it's really a pretty general a general mechanophore platform for the release of a wide variety of different functional molecules. And not only that, but by changing the substitution on our mechanophore, we can now really sort of pre-program into these materials the rate of release of that particular cargo molecule upon mechanical activation. So I'm going to switch gears just in the last little bit and tell you a little bit about our chemistry on the development of Naphthopyran mechanophores. So Naphthopyrans have been studied for a long time in terms of their photo switching properties. So this is a 3H Naphthopyran. Under UV light, it will undergo a ring opening reaction to generate these merocyanine dyes that are highly colored. And I'm just showing you here a particular selection where we can modulate the substitution at R and R prime positions and completely change the color of those merocyanines, for example, through simple substitution. And so it really, in our mind, is a really nice and modular platform for developing mechanochromic molecules and mechanochromic materials. And so many of you watching are probably perhaps unaware, but are probably already familiar with the chemistry of Naphthopyran. And that's because transitions lenses, right, the eyeglass lenses where you walk outside and they change color, are based on the active material in transitions lenses are actually based on Naphthopyran photochromes. And so the UV light from the sun triggers this ring opening reaction to generate these colored methionine dyes. And then you go back inside where there's no UV light and it transforms back into its colorless state. And so they're a very also commercially important class of molecules. In 2016, it was found that this same ring opening reaction of naphthopyran to generate these methionines can also be induced mechanochemically. And so this video shows an elastomeric piece of PDMS that covalently incorporates this naphthopyran. And as you apply tension to that material and stretch that out, that mechanical force induces that same ring opening reaction. Hopefully you saw that to change this material to this nice yellow color. Right? And so again, this sort of a demonstration of how these types of molecules, these mechanophores, really serve as molecular force probes, allowing us to visualize mechanical stress through this color change taking place really at the molecular scale. And so my group has looked at a lot of different things related to naphthopyran mechanophores. This particular slide shows how we've looked at how substitution on that naphthopyran scaffold can change things like the color that's produced in a mechanochromic polymer, and also the reversion rate of that methionine dye back to its colorless naphthopyran. Right? So we can make these yellow color changing naphthopyrans and these purple color changing naphthopyrans by installing this electron donating amines. And we can modulate their reversion rates by changing substitution both at the ortho position on one of these arenes, as well as the substitution on the naphthalene scaffold itself. Right? And we can use this chemistry to generate a wide variety of different mechanochromic properties with different mechanochromic materials with really sort of tailored mechanochromic properties. So that was looking at substitution. More recently, one of the things we've been interested in is looking at how the regiochemistry of naphthopyran influences its mechanochemistry. And so up to that point, all of the naphthopyran mechanophores that have been studied have this particular structure. This is what we refer to as a 3H naphthopyran. There's also 2H naphthopyran shown here, where essentially we've just flipped the orientation of that pyran ring about that naphthalene nucleus. And so we showed that 3H naphthopyrans will undergo this mechanochemical ring opening reaction to generate these yellow methionines that have pretty fast reversion. Whereas these 2H naphthopyrans are also mechanochemically active, but they generate methionines with bathochromically shifted absorptions, these red-colored methionines, and they revert relatively slowly. And so that's shown here in the solid state, again, through these elastomeric PDMS materials. And so now we can change substitution, we can change the isomerism of that naphthopyran mechanophore, and really have a number of different handles in which we can tune the mechanochromic properties of these types of polymers. So in the last just 5 minutes, what I want to tell you about those is some really unusual chemistry that we've discovered with naphthopyran. And that sort of, in contrast to what I just showed you in terms of just generating mechanochromic materials that have this binary sort of on-off response. One of the grand challenges that we've been thinking about for a long time is this idea of, can we design what I'll refer to as a multimodal mechanophore that is able to undergo different reactions under different amounts of stress or strain to generate discrete sort of visual cues. And so, just to sort of restate that, can we design materials or mechanophores that can distinguish between different stress states, right, through changes in absorption or changes in color? And so the sort of schematic conceptualization of this is just, can we have a material where you apply a small amount of stress or a small amount of strain, and it changes one color, say orange, and then we increase the amount of stress or strain applied to that material, and it changes a different color, right? If we can access this type of functionality, it would allow us to visually quantify, right, the amount of stress or strain a material is under, which would be really interesting. And so we first got sort of interested in this class of molecules called bis-naphthopyrans within this context. These have been reported previously to undergo this sequential ring opening reaction under UV activation, under UV light. And so under UV light, this bis-naphthopyran will first undergo one ring opening reaction to generate this open-closed species. And then upon additional UV irradiation, that second naphthopyran ring will open to generate this fully conjugated open-open species. Right? And so we see this change in color as a function of sort of temporal exposure to UV light. So we wanted to evaluate this in terms of its force responsive behavior. And in order to do that, my student Molly designed this bis-naphthopyran mechanophore and a library of polymers in which we just change the length of the polymer chain connected to this particular motif. Recall from the beginning of my talk that as we change the length of that polymer, under ultrasonication conditions, we change the amount of force that's delivered, or that polymer chain experiences. And so we hypothesized that changing the amount of force delivered to this mechanophore could change the distribution, perhaps, of these two different merocyanine isomers, and thus this material would experience a force-dependent coloration. And so to just sort of, as a background, we wanted to take those polymers that we had synthesized and confirm their photochemical characteristics, their photochemical reactivity, and that's what's shown here. If we take a 330 kilodalton polymer containing that bis-naphthopyran, and we subject it to UV irradiation, what we see first is the growth of a signal absorption peak at about 460 nanometers, which plateaus very quickly. So this reaction happens very quickly in the reaction. And that 460 nanometers is consistent with the formation of this open-closed merocyanine species. At later reaction times, upon photoirradiation, we then start to see the increase of an absorption peak at longer wavelengths that we're monitoring at 620 nanometers, which corresponds to the generation of this open species, right? So basically what I'm showing here is exactly what we expected based on the literature. This undergoes this sequential ring opening reaction upon UV irradiation. Now, what was surprising here was that if we take that same polymer and we mechanically activate it with ultrasound, qualitatively, you'll see that this spectra, the evolution of these spectra, look completely different. And so one of the things you'll notice is that even at early time points in this sonication, we're seeing that both of those species, that open-closed and the open-open species, are produced. And actually what we see is that the open-open species, that longer wavelength absorbing species, actually is formed faster than that open-closed species, right? In contrast to what we see in the photochemical experiment. Again, what we see is that if we change the molar mass of our polymer to now a relatively small polymer, 40 kilodaltons, which should experience lower forces, again, we see a difference in the spectral evolution between those two materials, suggesting that we are in fact seeing force-dependent changes in merocyanine formation. And so to investigate this a little more quantitatively, what we did was we looked at those temporal profiles under sonication, looking at the absorption at 460 nanometers compared to that at 620 nanometers. And we can just fit these to simple first-order kinetics, and then we can project this out to measure what the projected plateau value is at each of those wavelengths. And so importantly here, this is the plateau value I identify as a B value, so B at 460 nanometers, B at 620 nanometers. The important thing to note here is that the ratio of these two values describes the distribution of these two different merocyanine products. And so what we see actually is that if we look at the ratio of those B values, right, the distribution of those merocyanines is predictably changed as a function of the amount of force that we apply to that mechanophore. Right, so at low forces, low molar masses of our polymers, we see primarily the generation or an absorption spectrum that's dominated by this open-closed species, and at much higher forces, we see an absorption spectrum dominated by the formation of this open-open species. And we can see that in these photographs here, right? Under low forces, we see the generation of this sort of yellow-orange color, and at high forces, we see this purple color. And so, how do we explain this? So again, if we look at the mechanism of the photochemical reaction, again, this undergoes a sequential ring opening reaction, where under UV light, we get one of those naphthopyrans to open, generating this open-closed species. And then upon further UV irradiation, we get the second ring opening reaction to occur to generate this open-open species. And both of these processes are thermally reversible. What we see in the mechanochemical reaction is that the mechanism actually changes. And so our data is consistent with the direct dual ring opening under force of both of those naphthopyrans to directly convert our bis-naphthopyran into this open-open species, the rate of which is dependent on the force applied to that particular molecule, right? And we're generating now this open-closed species through the thermal reversion of this particular purple-colored merocyanine, right? And so by changing the amount of force applied to bis-naphthopyran, we're modulating the rate of formation of this species relative to the rate of formation of our open-closed species. And this is essentially sets up a dynamic equilibrium in which we can change the distribution of those two products as a function of force. So in my last two slides here, I want to tell you where we're sort of going with this and some really recent results from the lab, again, related to this idea of the development of these multimodal mechanophores. Related to that bis-naphthopyran that I showed you are these naphthodipyran compounds. And so these are a little bit different because we have a single naphthalene core, and fused to that are two pyran rings. And so what's been known in the literature about naphthodipyrans is that upon UV light, you can only get one of those pyran rings to open to generate these monomerocyanines, and you never get both of those rings to open under photochemical activation. Right, so these dimerocyanines are not formed. We hypothesized that actually using mechanophores, we may actually be able to get both of those rings to open, leading to some unusual and potentially interesting chemistry. And in fact, what we've done is we've designed this particular naphthodipyran and shown that upon incorporation into polymeric materials, indeed UV photoirradiation generates a single produces a single ring opening reaction to generate these monomerocyanine dyes. Whereas if we take that same polymer and we subject it to mechanical force under ultrasound, we can indeed open both of those pyran rings to generate these really unusual dimerocyanine compounds. I'll just note briefly that the structural attributes of this mechanophore, we've installed these terminal pyrrolidine groups because they allow for the use of a BF3 as a Lewis acid to essentially irreversibly trap the merocyanine dyes that are produced. And so it was just sort of an experimental feature that allowed us to actually better identify and characterize the formation of these species. So I'll just show you the results. If we take that naphthodipyran containing polymer and we irradiate it with UV light, we see the growth of a peak at about 620 nanometers, a single absorption feature, and this nice blue-colored species. Right? And this is consistent with the formation of this monomerocyanine compound. However, again, if we take that same polymer and we subject it to ultrasound, we see the generation of a completely different species, here with an absorption going way out into the near-infrared region, which is consistent and which we've characterized to be this dimerocyanine, in which both of those pyrans have opened to generate this particular compound. One sort of interesting thing of note is that in contrast to what we saw with bis-naphthopyran, we actually see a different sort of temporal evolution of these two species as a function of sonication time. And so early in the reaction, we see the growth of this monomerocyanine species, the accumulation of that monomerocyanine, and then only at later reaction times do we see the accumulation of this dimerocyanine species. And so in this particular case, it's really that the color of that solution is really reporting on the sort of temporal exposure of that material to ultrasound, which is sort of an interesting feature. Okay, so I'm going to end there. Hopefully, I've told you about several different sort of flavors and programs within my lab. This idea of how we can design mechanophores based on this mechanical gating strategy to achieve materials with very different functional properties, from mechanically gated photo switching to the mechanically triggered release of covalent small molecules. And then finally, how we can use Naphthopyran to develop mechanochromic materials and also explore some really interesting and unusual reactivity that comes out of Naphthopyran under mechanical activation pathways. So I need to thank my group. I've tried to highlight the folks that were responsible for the projects that I talked about throughout my talk, but I have a phenomenal group of graduate students and postdocs and undergraduates that work with me, none of which the results I showed with you would be possible without their efforts. So they're really the ones that did all of this work. And then finally, I'll thank the funding agencies that have financially supported our work, and I'd be happy to take any questions.
Thank you so much for that presentation. We have quite a few questions. All right, let's start with using ultrasonication. What is the magnitude of the force being applied, and what are the strain rates?
That's a really good question. So I can answer the second one more concretely. The average strain rate is on the order of about 10 to the 6th per second. And so we're working on sort of the microsecond timescale with respect to strain rates under these conditions. The magnitude of force is less certain. We're certainly operating in the nanonewton force regime, right? Because we're breaking covalent bonds. And so my personal feeling actually is that we're probably operating, depending on the length of the chain, I think we're probably operating more or less in the range of about 2 to potentially 5 nanonewtons of force. some people don't think that we're getting up to about the 5 nanonewton range, but I think that there's evidence. We know that at these timescales, that to break a covalent carbon-carbon bond requires about 5 nanonewtons of force. And we know that if you have just a polymer chain, we know that that polymer chain will cleave. And so that, to me, tells us that we're operating, in that particular regime. So, 2 to 5 nanonewtons is what I suspect is the regime. It's hard to quantify, though, actually.
Thank you. Another question. What were some of the challenges that drove you to generate your second generation photoswitch design?
Yeah, that's a great question, and I really did gloss over that. It was really so the synthesis of that first generation Diels-Alder adduct was really challenging, and one of the challenging aspects was the fact that we had to, one of our intermediates in that synthesis was a cyclopentadiene, but we had no control over the regiochemistry of that cyclopentadiene. So when we formed a variety of different tautomers, and then we had to use that mixture to generate our Diels-Alder adducts. And so from that reaction, we actually generated 6 different Diels-Alder diastereomers, where we had to use, actually in this case, preparative supercritical fluid chromatography to isolate our material of interest. And so, thinking about this was not, it got us to the material and allowed us to sort of investigate the chemistry, but in terms of functional, mechanophore, it wasn't a great approach. And so we went back to the drawing board. I'd say the second thing to that actually is also that it really wasn't very modular. So if I talked about how we could change the substitution on that masked photo switch. In order to do that in our first generation system would have required a tremendous synthetic effort. And so we wanted to basically go back to the drawing board and develop a much nicer, better synthesis of that mechanophore that also allowed us to change the sort of functional properties at a late stage in the synthesis. So installing different arene functional groups that would allow us to have access different types of photo switches upon this mechanically gated reaction.
Thank you. Here's another. Have you ever compared the mechanochemical activation of your photo switches in the solution phase under sonication to the polymer activation in the bulk solid phase? Are they different, or how are they similar?
Yeah, that's also a good question. To be honest, we haven't explored to nearly as great of an extent the activity in the solid state. So what we showed in that chemical science paper was that it works. We can achieve mechanically gated photo switching in solids, but beyond that, we really haven't quantified the activity very much, or the chemistry very much in solid state materials. It becomes more challenging to do that, which is one of the reasons why we've primarily focused on, especially in the development of new chemistry, using ultrasonication methods to evaluate and characterize that chemistry. It becomes more challenging to just do that same type of chemical characterization when you're working in solid state materials. And so for that reason, we haven't really gone down that path as much. And so I can't say much more to that.
All right. I'll switch gears a little bit. Regarding the flower-shaped stamps, we noticed that near the center of the flower, the color was less distinct or more faded. Is this because there's a lower force near the middle for some reason?
I think it's just based on the shape of that particular stamp and how force is actually distributed in the material when we compress it. So I think it's really just based on the geometry of the stamp that we used. I think that we could optimize this so that we didn't have this sort of middle region that didn't undergo as much stress or strain as the sort of peripheral region of that stamp. But I think it's really just stamp geometry that isn't optimized in that particular case.
Great. All right, let's do lots of questions. Let's do just one more. How sensitive are your molecular force probes? What's a reasonable force range for these sensors? And does the force range change depending on the polymer the mechanophore is incorporated into?
So the force is really going to be controlled by the reactivity of the mechanophore. And so the range again sort of gets back to one of the earlier questions, which is, what is the force range that is experienced under, particularly in ultrasonication conditions? Our best estimates from this come from computation, unfortunately. So there are a number of studies where we can use single molecule measurements, things like single molecule force spectroscopy, to really accurately measure the force at which these reactions take place. None of those experiments have been done on say, Naphthopyrans or the molecules that I've discussed today. But we know from those experiments that the forces range, say, like spiropyran is one of the most active mechanophores that's been studied. So single molecule measurements on spiropyran reveal a ring opening reaction that occurs on the order of about 300 piconewtons. It's actually very reactive. And we also know from single molecule force spectroscopy experiments that some mechanophores will react upwards of about 2.5 nanonewtons, for example. So that's sort of the range that we're working with these covalent type mechanophores. But I don't have a great estimate for some of the reasons that I discussed related to that first question on exactly what forces these mechanophores are reacting at, other than from those Fmax values that we get from COGEF, which are good relative predictors of reactivity, but not necessarily great, predictors in terms of the accuracy of the magnitude of that value. But we're working within a general range of let's say 2 to 5 nanonewtons. I would say is a reasonable range.
Got it. All right. Well, thank you so much again for your presentation and for being part of the seminar series.
Thank you.
Thank you, Professor Robb, for that outstanding presentation. And thank you so much for joining us. Just a reminder, as part of the seminar series, we've had a great group of speakers so far, and you can watch any of these anytime on the YouTube channel. We also have a good group of speakers in the upcoming months, and we hope that you will join us for all of them. Thank you again.